Hi everyone, and welcome back to day 3 of the OHBM Open Science Room program. Today our theme is Past, Present, Future of Open Neuroimaging, and this session is about demonstrations, both process demonstrations and software demonstrations. Just to check in again, you can see on our website, the link is here on the slide. You can find more information about our speakers and about the schedule. You are also very welcome and encouraged to share what you experience here in the Open Science Room on Twitter, and we've provided some hashtags there. You are also very welcome to join the discussion about the Open Science Room in general on Mattermost, for which we've provided a channel name there, and also for this theme and this talk, or these talks in particular, on the OSR Future channel in Mattermost.
So in these demonstrations, we'll be looking at the tools that we are using and developing, and specifically where they're heading in the future of our work. We'll have 4 talks of 7 minutes each, and then there'll be 20 minutes for Q&A with speakers, live Q&A. And some, but probably not all speakers will be available in this time zone that you're joining us from. So any questions that can't be answered right now, we will collect them and pass them on to the speakers for them to be answered.
So the platform that we'll use for Q&A or questions and answers is Crowdcast. So you can add your text-based questions there and upvote them, and we'll get them to the speakers for the live Q&A session. If you can't use Crowdcast, you're welcome to also post your questions in the OHBM platform, or join the discussion on Mattermost. We'd love your feedback for the OSR in general, but also for the speakers. So please fill in the polls that we've created for the session. And also remember to treat each other with kindness and respect. So we've all agreed to abide by the code of conduct when we registered for this event. And we ask you to, if you see or experience any violation of this code of conduct, to report that following the structure that's given in the code of conduct, which is available on the OHBM website.
Now it's my pleasure to introduce our speakers for this session. We have Fabio Ferreira, who will be discussing the Women in Neuroscience repository. Marijn van Vliet will be giving us a status update on NME Python, followed by Srinivas Rachakonda, who will be introducing new features and developments in the group ICA Toolbox. And finally, Ariel Rochem will be giving the INCF online training suite introduction and look through, which is a resource to advance, enable, and education in brain data science. So thanks again for joining us for this session, wherever you're joining from, and we look forward to these talks and then interacting with you in the Q&A at the end of the session. Thank you very much.
Hello everyone, I'm Fabio, and I'm doing a PhD in computer science at UCL, London, and today I'm going to talk about the Women in Neuroscience repository, or just WinRepo, and how it can be used as a powerful tool to promote gender equity in neuroscience. But before I start, I would like to say that I hope you are doing well, and wherever you are in the world, you are safe. So what does WinRepo bring to the community? As highlighted by different initiatives and academic papers, gender balance in neuroscience is still work in progress. And one key aspect that still needs to be addressed is the underrepresentation of women at conferences and other academic events. To tackle this issue, WinRepo was created, and it provides a comprehensive search engine to find women profiles, as well as a recommendation system. I will show you how you can search in WinRepo in a moment, but before I do that, I want to talk about a bit about of the history of WinRepo. So Jessica, at the beginning of 2018, after seeing another academic event full of men speakers and only a single woman speaker, she decided to start collecting women profiles in neuroscience by sharing a Google form. And a few days later, together with Greg, they created a website already with 250 profiles. So after a few months, the WIN Repo was released at OHBM 2018 and was well received in the community. And because of this, some volunteers joined the initiative and to form a board. And since then, they have been working to increase the visibility of the website and also the impact of the initiative. Nowadays, the WIN Repo has already more than 1,400 profiles, as you'll see in a moment. So now, as I said before, I want to run a small demo. I'll show you how to register a profile on the website, how to search profiles using specific keywords or filters, and how to submit a recommendation. To do that, I need to go to the website. So this is the home page. You can search profiles by using keywords already here, or you can view the repository by click this button, create a profile by click this one, or recommend someone by clicking this one. We have this nice feature where 6 random recommendations that have been previously submitted to the website, they are shown here. And here you can also see the number of profiles in the repository already. We can see that we have a nice distribution of the levels of seniority. And also in this map, you can see the geographical distribution of the profiles, which is already a considerable one. And finally, if you want to help us raise awareness on this issue, please download this slide that we provide here and insert it at the end of your presentation whenever you go to an academic event. So right now I'll show you how to create a profile on the website. You can click it. You can create it by clicking here or here, and then you have a form that you need to fill in. I'm creating a fake profile just to show you how to create it. So you need the full name, institution, country of residence, positions. Then you need an email. You can also add a web page, but I'll not do it now. You can add your date you obtained the PhD. Let's do it. And then you can also add some fields of research. Brain for my instruction, you can choose whole brain, for instance, MRI, univariate and multivariate, memory, for instance, in the domain, and language. And finally, you can also add some more keywords, for instance, machine learning. Then you submit the profile. You can see that the profile was created successfully, and all the information that you just fill in is provided here. And then if you want to write a recommendation to this person, you can click here. You can also click in this button here as well. So the name will appear here, the name of the person that you want to recommend. Then you need to add your name, position, your institution. Guys saw one of our talks, and I have one example of recommendation here. You can edit here, and then you submit it. And finally, you can see that on the profile, you have one recommendation now. And now I'll show you how to visit the repository. You can click here, and then you will have access to all the entries of the repository. Now you can use keywords to search for any profile. I can click, MRI, and then you do search. You can see our profile here, we just created, and you have many more, more than 700 entries. And then you can use this filter under represented countries. You can see now Angola is one underrepresented country, so that's why our profile is here. You can also filter by senior position. And you can see that still we have, in this case, 14 entries. You can add more keywords if you want, for instance, language. Search, you see that you have now just 3 entries. And yeah, And that's it. So I just want to show you a few take-home messages. So first of all, so Women Neuroscience, please sign up on the website. Organizing committees, use the website to find women profiles. Everyone, please spread the word and submit recommendations. And finally, we are looking for volunteers. I hope the site will be provided, and you can find the form here. I'll show you in a bit the form. And we during the conference we will also be sharing our this form in our Twitter. You can follow us on Twitter and Facebook. And and finally, you have also some useful links here. The first one is for the website, and just to let you know that the Win repo is publicly available, and it can be found in the GitHub repo using this link. And if you want to collaborate with us, please let us know. And I'd like to finish by thanking everyone that has been directly or indirectly involved in this initiative. So thank you very much, and I hope you have a nice day.
Hi all, I'm going to give a demonstration about Group ICA Toolbox, new features and developments. So the highlights that I'm going to cover are standalone gift, automated ICA or reference-based ICA, and some dynamic functional connectivity highlights. See, here is a brief introduction on GIF Toolbox. GIF Toolbox is used to run single subject as well as group level ICA on fMRI data. It's an open source MATLAB toolbox, and there are about 19 algorithms implemented like Infomax, Fast ICA. There are options to run reference-based ICA or automated ICA, where you can give priors as spatial templates, and GIFT can run very, very large analysis using MPOWIC and subsample time PC algorithms. Some of the tools in GIFT are like Mancova, where you can run multivariate and univariate tests given the covariates, and it is computed on the ICA output. And dynamic connectivity, where you can run temporal and spatial dynamics on the ICA or IVA components. Noise cloud. Noise cloud is a utility which can detect noise or component from the data. And the features it uses are spatial maps and time courses. There is a remove artifacts, where you can remove artifacts from the data and run an analysis like GLM. And there are options to remove bad or outlier subjects from the data, from the analysis using the generate mass tool. So also spatial temporal regression, basically it's a utility where you can give like prior ICA maps and unknown data set, and you can estimate subject maps and time courses. Here is a group ICA framework. I simplified it a bit. So basically you have time by voxels on the left hand side for each subject. It's stacked across time dimension, and after ICA you get time by components, component by voxels. So this is a simple example where I'm not covering the PCA whitening and de-whitening matrices, just to give an idea of how group ICA works. Standalone gift, basically gift is built in MATLAB, so you can run without MATLAB by compiling it to Windows and Linux operating systems. Also, there are options to use Docker for group ICA, and you can also run GIFT without MATLAB using Python wrappers through NiPy interface. So here is a small picture on your top left hand side. You can see downloaded standalone GIFT. First, you need to install the MATLAB runtime and set environment variable, which is shown on the right hand side. And after setting environment variable and executing group IC application, you get the Toolbox, which it has all the features when it is run with MATLAB. So here is a screenshot of Docker link. Basically, it's on GitHub. You need two files like build.shell and run.shell. Build.shell builds a Docker image, and run.shell you can customize the inputs and outputs on your local directory to be mapped onto the container file. So all the instructions are there in the readme.txt file in the GitHub link. You can also invoke standalone Gift using an IPipe interface. You can install the package using pip install and run Gift in a Python environment. Automated ICA or reference-based ICA is basically you give spatial priors as templates to a subject data, and using the spatial priors as constraints, you get time courses and spatial maps. So here is a screenshot of what automated ICA does. So when you click on setup analysis on your left hand side, you can see a small window pops up on the right hand side where there's a reference based ICA. And when you click on that, there is a GUI which shows three panels: data, algorithm, and template selection. So template selection is where you give priors. So here are the results. The composite ortho views of the spatial maps and functional network connectivity represented as a connectogram. Dynamic connectivity toolbox. You can study temporal and spatial dynamics using this toolbox. So it is when you click on dynamic functional connectivity on the Gift, you get like a toolbox which has temporal and spatial panels. So temporal panels, most of the these analysis work on the ICA output from Gift. The exceptions are ROA based DFNC, which works on the bold fMRI output, and there is this spatial DFNC, which works on the raw data as well, using IVA on the partition data. Here is an example of a spatial quantum toolbox. When you click on the toolbox, you can see an option to select components, and after you select the component, there is a pairwise correlation computed between the time course of the component and the bold fMRI data. So what you get is a uh, spatial map, that is windows by voxels for each subject, and you can do like different types of post processing, like running k-means on the output. And on the right hand side, you can see the spatial states of the DMN. Spatial dynamics hierarchy is typically used when you have a higher model order. There is a component splitting. So you can group components as one network, and then like you can enter different networks like default mode or visual. And what happens is time courses of the components, default mode components, are multiplied with the spatial components to get the data set. It is like time by voxel, and it is stacked across subjects, and you can do like k-means. And here is the result of the spatial states obtained for the default mode network. So here are some references. You can download Gift when you go to trendcenter.org, and there is Gift Docker toolbox videos are there, which are describe in detail about the DFNC, and there are some templates also you can download, and main papers that are described in this tutorial are also there. Thank you for listening.
OHBM 2020 Open Science Room. I'm here with an update on the MNE-Python project. MNE-Python is an advanced analysis package for time series neural imaging data, such as MEG, EEG, ECOG, and FNIRS. Whatever your needs are for pre-processing your data, chances are good that MNE-Python can do it. Let's take a look at some of the things it can do. We can annotate bad channels, bad segments, bad epochs, and MNE-Python now contains a complete implementation of Megan's max filter. You can compute independent components, so you can get rid of eye and heart artifacts, cut epochs, annotate them with metadata, and study evoked activity any way you like. And when it's time to localize the actual sources in the brain, perform co-registration between your sensors and an MRI scan. Construct BEM models, define a source space, and compute the lead field. And when it comes to the actual source localization, there are so many ways to do it. Use cluster permutation testing for group level statistics, and so much more. Now, let's take a look at what we've been adding since last we saw each other at OHBM. We've implemented the missing pieces to make our open source max filter implementation just as good as the commercial Megan version. And this means we now support continuous head position tracking and correct for it. You can use max filter for automatic bad channel detection of your MEG data. Our co-registration GUI has gotten some love as well. It will now attempt to automatically detect the MRI fiducials. It doesn't always work, but when it does, your co-registration can be performed completely automatic. Our topomap plots are looking better than ever. We've overhauled how we detect the head position and deal with subsets of sensors. And speaking of sensors, you can now visualize sensor-to-sensor connectivity in 3D. We also support more file formats than ever, including curry files, the new NYU 2019 kit system, and we can directly import independent components straight from EEG Lab. We're also faster than ever, with speed improvements all over the codebase. And finally, there's one more thing. A completely new visualizer for source activity based on PyVista. You can scroll smoothly through time, or just press the spacebar to play it as a movie. You can quickly adjust color thresholds and click on any vertex to get a plot of the time course. But MNE-Python is more than just a fantastic piece of software. Above all, we are a community of scientists, sharing our expertise so that every one of us benefits. Every time we solve a problem or find a better way to do things, all of our analysis pipelines improve a little. Our GitHub page is always buzzing with issues and pull requests. Come take a look and see what we're working on next. Even better, please join in! We encourage everyone to file new bug reports and contribute pull requests with new features. Alex and Eric are usually the first ones to respond. Become a contributor to MNE-Python and you will improve your programming skills, you will make new friends, and you will find that when you put in some effort to help other people, others will gladly put in effort to help you. You don't have to solve every problem by yourself. Come join us.
Hello there. My name is Ariel Rokem, and I am a data scientist at the University of Washington. And today I would like to tell you about work that I've been doing together with the Training and Education Committee of the International Neuroinformatics Coordination Facility, INCF, on neuroeducation without borders. And I should give most of the credit actually to INCF, and particularly to Matthew Abrams, who's the deputy director. And those really are the people who have done the work here, which I will be representing. So first of all, you might be wondering what the International Neuroinformatics Coordination Facility, or INCF, is. INCF is a standards organization for open and fair neuroscience. And so INCF works on developing standards and supporting standards and endorsing standards, working groups and special interest groups working on a variety of topics around neuroinformatics, the use of computing in neuroscience. And then, in addition, there's a whole arm of INCF, which I will talk about today, devoted specifically to training, providing educational resources for a global neuroscience community, both through the online resources that I'll show today and through in-person courses. And then the INCF holds the INCF Assembly, which is a once annual conference, such as very similar to OHBM in scope and scale, that is devoted specifically to neuroinformatics. And so today I'll talk about the training activities, and I'll talk in particular about the online resources that INCF has created for training. Neuroinformatics requires a variety of different skills, both knowledge about neuroscience topics, but also technical, analytic, computing kind of skills, and in tandem also tools from library information science and quantitative skills. And so the training resources needed in neuroinformatics are actually quite broad. The first kind of resource that I'll talk about, and the one that I'll devote most of my presentation to, is Training Space. Training Space is a website that serves as an online hub to make neuroscience educational materials accessible to the global neuroscience community. So it provides users with access to multimedia educational content from existing courses, from conference lectures, and from lab exercises. And then it organizes these resources, these educational content, into study tracks that facilitate self-guided study. The website, Training Space, provides tutorials on tools and on other open science resources for neuroscience research, and connects to a Q&A forum. In addition, there's a neuroscience encyclopedia, or data discoverability portal, that provides users with access to many publicly available data sets, so that the knowledge that is acquired can then immediately be applied to data sets and other resources like literature references and scientific abstracts. So in terms of the content is developed in partnership with many different organizations, including OHBM, and the videos are provided by those organizations, and the content is provided by those organizations and others, and are on a variety of topics: general neuroinformatics, computational neuroscience, clinical neuroscience, and general neuroscience, data science, and computer science. And then there are created study tracks that focus specifically on computational neuroscience, on neuroinformatics, and on neurobiology. So this is what it looks like then. You go into the website and you can find the top courses within a particular topic like computational neuroscience. You can browse courses on particular tools and resources, for example, the virtual brain. And these are organized into these study tracks. So here's a study track then that contains many different courses. in addition, you can focus specifically on a particular course. For example, if you'd like to take the HBP education program, the Human Brain Project, the European Human Brain Project, you can focus specifically on their courses. And that gives you then access to these kinds of videos, slides, and other resources. If you focus in contrast on tools, then you can get access to various things like the papers, but also the code that is part of the resource. And then links to GitHub where the code lives. Now, in complement to these courses, there's Knowledge Space. This is a community-based encyclopedia that contains many different entries about different topics in neuroscience. For example, a topic such as neuron, and those are interlinked together, and link out to various other resources such as data sets and so on. And this works as a kind of Wikipedia focused on neuroinformatics, where you can search and find the different topics that you're interested in exploring. Finally, the last kind of resource that I like to talk about is a question and answer forum. This is called Neurostars. Neurostars is a great project. It's a question and answer website that you should really all be using all the time, where you can ask questions about various neuroinformatics topics. The website right now has a very strong emphasis actually on neuroinformatics in human brain mapping. So this is a great place to ask about, for example, tools like fMRI prep or different kinds of human neuroimaging data or diffusion MRI and so on and so forth. As of 2018, it had about 700 registered users, but that community is rapidly growing and with multiple visits from users all over the world. And so finally, I'd like to acknowledge the people who've worked on this. So this is the team, the training space development team at INCF. I'd like also to acknowledge the INCF Training and Education Committee that I am part of, and you can see the members of at one of our meetings. And then we'd like to thank, of course, the INCF community for providing the resources, the multimedia content, and the courses that are provided through these resources. Thank you.